Dear students, today we will discuss about the fatty acid biosynthesis. Before dealing with the topic, that is the biosynthesis of these fatty acids, we shall be aware about the fact that what are these fatty acids. When we define these fatty acids, we define them as a class of compounds containing a long hydrocarbon chain and a terminal carboxylate group. These fats and oils belong to the general class termed as the lipids, which are a structurally diverse group of hydrophobic compounds that are soluble in organic solvents such as chloroform, methanol, and are highly insoluble in water. These fats and oils exist mainly in the form of triacylglycerols, in which these fatty acid molecules are linked by ester bonds to the 3 hydroxyl groups of glycerol. These fatty acid chains often differ in length, categorized as short, medium, or long fatty acid chains. We call the short chain fatty acids as those fatty acids which have aliphatic tails fewer than 6 carbons long, for example, butyric acid. Medium chain fatty acids are those fatty acids which have the aliphatic tails of 6 to 12 carbons long, which can form medium chain triglycerides. Long chain fatty acids have the fatty acids with aliphatic tails longer than 12 carbons long. Very long chain fatty acids is another class which have aliphatic tails longer than 22 carbons long. Now, coming up to the functions of these fatty acids. Fatty acids have 4 important functions. Firstly, fatty acids function as building blocks. These fatty acids are the building blocks of phospholipids and glycolipids, which are the constituents of the cell membranes. Fatty acids as targeting molecules. Fatty acids are attached to many proteins. In this way, proteins are directed to their appropriate place across the membranes. Now, fatty acids as fuel molecules. We know that fatty acids are stores of triacylglycerols, and these triacylglycerols are used as fuel molecules. Now, fatty acids as messenger molecules. Products of these fatty acids functions as hormones and as intracellular messenger molecules. Now, coming up to today's topic, that is fatty acid synthesis. When we define this process, we generally define it as the creation of fatty acids from acetyl coenzyme A and malonyl coenzyme A precursors through the action of enzymes which we call generally as fatty acid synthase. This fatty acid synthesis starts with the carboxylation of the acetyl coenzyme A so as to form a malonyl coenzyme A. Therefore, the input to this fatty acid synthesis is acetyl coenzyme A. This carboxylation of the acetyl coenzyme A or the malonyl coenzyme A synthesis takes place in 2 steps. First, the acetyl coenzyme A is carboxylated so as to form a carboxybiotin intermediate at the expense of hydrolysis of molecule of ATP. That means it is an energy requiring step. The activated carbon dioxide group in this intermediate, that is the carboxybiotin intermediate, is then transferred to acetyl coenzyme A so as to form a malonyl coenzyme A. The synthesis of this malonyl coenzyme A is catalyzed by the enzyme acetyl coenzyme A carboxylase. This enzyme, that is the acetyl coenzyme A carboxylase, it is a multi-enzyme protein. For example, in bacteria, acetyl coenzyme A carboxylase has 3 separate polypeptide subunits. In animal cells, all these 3 activities are a part of a single multifunctional polypeptide, while as the plant cells contain both types of the acetyl coenzyme A carboxylase, that is the type found in the bacteria as well as the type found in the animal cells. However, it should be noted that in all the cases, enzyme prosthetic group is biotin. Now, after the initial malonyl coenzyme A synthesis, the long carbon chains of fatty acids are then assembled in a repeating 4-step sequence. That is, the condensation, reduction first, dehydration, and the reduction second. And with each passage through this cycle, through these 4 steps, the fatty acyl chain is extended by 2 carbons. All the reactions in synthetic process are catalyzed by a multi-enzyme complex, fatty acid synthase. This fatty acid synthase is the main enzyme that is responsible for the synthesis of these fatty acid long chains. Fatty acid synthase is a complex of enzymes, not a single enzyme, that adds C2 units to a growing fatty acid chain. Fatty acid synthase is a multi-enzyme protein consisting of 7 separate polypeptides. Proteins act together to catalyze the formation of fatty acids from acetyl coenzyme A and malonyl coenzyme A precursors. This fatty acid synthase has a large diversity. For example, I would like to tell you that in bacteria and some plants, 7 active sites for fatty acid synthase reside in 7 separate polypeptides, whereas in yeast, the 7 distinct active sites reside in 2 large multifunctional polypeptides with 3 activities on the alpha subunit and 4 on the beta subunit. However, in vertebrates, a single large polypeptide contains all the 7 enzymatic activities. Now, this fatty acid synthase receives the acetyl and malonyl groups. During the fatty acid synthesis, first, the acetyl group of the acetyl coenzyme A is transferred to the cysteine group of the beta ketoacyl ACP synthase. And this reaction is catalyzed by the acetyl coenzyme A ACP transacetylase, an enzyme of the fatty acid synthase. The second reaction is the transfer of the malonyl group from the malonyl coenzyme A to the SH group of the ACP. And this reaction is catalyzed by the malonyl coenzyme A ACP transferase. And this enzyme is also a part of the complex. Thus, in the charged fatty acid synthase complex, the acetyl and the malonyl groups are very close to each other and are activated for chain lengthening process. After this initial attachment of acetyl and malonyl groups with fatty acid synthase, the first reaction in the formation of fatty acid chain involves condensation of the activated acetyl and malonyl groups to form a acetoacetyl ACP, which remains bound to the ACP through the phosphopentadiene group. Simultaneously, a molecule of carbon dioxide is also produced. In this reaction, which is catalyzed by beta ketoacyl ACP synthase, the acetyl group is transferred now from the cysteine group of the enzyme to the malonyl group, thus becoming a methyl terminal 2-carbon unit of the new acetoacetyl group. Now, the acetoacetyl ACP formed in the condensation step undergoes reduction of the carbonyl group at the carbon number 3 so as to form a D-beta hydroxybutyryl ACP. And this reaction is catalyzed by the ketoacyl ACP reductase. Here, the electron donor is NADPH. Now, after the reduction of this carbonyl group, the elements of water are now removed from carbon number 2 and carbon number 3 of the hydroxybutyryl ACP so as to yield a double bond in the product, which is the butynyl ACP. The enzyme of the fatty acid synthase that catalyzes this dehydration is hydroxyacyl ACP dehydratase. Finally, the double bond of the trans-butynyl ACP is reduced. That is, it is saturated so as to form a butyryl ACP by the action of enoyl ACP reductase with NADPH as an electron donor. This enoyl ACP reductase is also a part of the fatty acid synthase complex. Production of the 4-carbon saturated fatty acyl ACP completes one pass through the fatty acid synthase complex. The butyryl group is now transferred from the phosphopentadiene group of the ACP to the cysteine group of the ketoacyl ACP synthase, which initially bore the acetyl group. To start the next cycle of the 4 reactions that lengthens the chain by 2 more carbons, another malonyl group is linked to the now unoccupied phosphopentadiene group of the ACP. Condensation occurs as the butyryl group, acting like the acetyl group in the first cycle, is linked to the 2 carbons of the malonyl ACP group with concurrent loss of the carbon dioxide. The product of this condensation is now a 6-carbon acyl group, which is covalently bound to the phosphopentadiene group. Its beta keto group is reduced in the next 3 steps of the synthase cycle to yield the saturated acyl group, exactly as in the first round of reactions, but in this case forming the 6-carbon product. 7 cycles of this condensation and reduction produces the 16-carbon saturated palmitoyl group, which is still bound to the ACP. Chain elongation by the synthase complex, that is the fatty acid synthase complex, generally stops at this point, and free palmitate is released from the ACP by hydrolytic activity in this complex. Now, if we talk about the energetics up to this point, I would like to recall that the synthesis of this palmitate requires the input of 8 molecules of acetyl coenzyme A, 14 molecules of NADPH, and 7 molecules of ATP. In certain plants, for example the coconut and the palm, chain termination occurs earlier, and up to 90% of the fatty acids in the oils of these plants, that is the coconut like the palm, are between 8 and 14 carbons long. No doubt, small amounts of the longer fatty acids such as the stearate are also formed. Once the long chain acids have been produced, for example the palmitate, they can be subjected to modifications like the elongation and desaturation. We will discuss about these modifications. Let us discuss first about the synthesis of long chain saturated fatty acids. Palmitate, the principal product of the fatty acid synthase system, is the precursor of the long chain fatty acids. It may be lengthened to form the stearate, which is an 18 carbons long, or even a longer saturated fatty acids by further additions of acetyl groups through the action of fatty acid elongation system present in the smooth endoplasmic reticulum and in the mitochondria. Thus, the synthesis of these long chain saturated fatty acids requires a separate enzyme system which we call as fatty acid elongation system, not the fatty acid synthase. The more active elongation system of this endoplasmic reticulum extends the 16-carbon chain of the palmitoyl coenzyme A by the 2 carbons, forming citrate coenzyme A. Although different enzyme systems are involved and coenzyme A rather than the ACP is the acyl carrier in the reaction, the mechanism of elongation in the endoplasmic reticulum is otherwise identically to that of the palmitate synthase, and donation of the 2 carbons by malonyl coenzyme A followed by the reduction, then dehydration and a reduction to the saturated 18-carbon product citrate coenzyme A. Now we will discuss about another modification, that is the synthesis of desaturated fatty acids. As all of we are aware about the fact that lipids of all higher organisms contains an appreciable quantities of polyunsaturated fatty acids with methylene interrupted double bonds. In higher plants, the number of the double bonds in fatty acids only rarely exceeds 3, but in algae and animals there can be up to 6 double bonds in a single fatty acid chain. The introduction of the double bonds into the fatty acid chain relies on the activity of desaturases, which are actually mixed function oxidases. That is, a separate enzyme system is required to introduce a double bond into a fatty acid chain. These desaturases work via an aerobic mechanism, with oxygen being reduced by 4 hydrogen, 2 from the substrate fatty acid and 2 from the reductant that is used. In other words, 2 electrons pass from the NADH to the desaturase and 2 electrons are extracted from the fatty acid as the double bond is formed. Thus, there is a 4-electron reduction of oxygen so as to form 2 molecules of water as a fatty acid is oxidized to form a double bond. It should be noted that these desaturases are specific for specific positions of the double bond. For example, palmitate and stearate serve as precursors of the two most common monounsaturated fatty acids, that is palmitoleate and oleate. Palmitoleate is a 16 carbon long and oleate is 18 carbon long. Both of these fatty acids have a single double bond between carbon number 9 and carbon number 10. However, it should be noted that mammals cannot introduce the double bond beyond the carbon number 9 and have only desaturases which can introduce the double bond at carbon number 9, carbon number 6, carbon number 5 and carbon number 4. Many of these unsaturated fatty acids, nonetheless, for example, the linoleic acid, which have a double bond at carbon number 12, or linoleic acid, which have a double bond at carbon number 12 and carbon number 15. These are present in the mammals. Therefore, mammals must take these fatty acids into their diet from the plant oils. Since the mammals do not have the desaturases that can introduce the double bond beyond the carbon number 9, have to be supplied in the diet, and these fatty acids are called as essential fatty acids. Now, we should talk about the regulation of the fatty acid biosynthesis. When a cell or an organism has more than enough metabolic fuel to meet its energy needs, the excess is generally converted to fatty acids and stored as lipids such as triacylglycerols. The first reaction of the fatty acid synthesis, catalyzed by the acetyl coenzyme A carboxylase, is thus an important site of regulation. Acetyl coenzyme A carboxylase enzyme, which initiates the fatty acid synthesis process, is highly regulated. For example, this mammalian enzyme, that is mammalian acetyl coenzyme A carboxylase, it is inactivated by phosphorylation, which is favored by glucagon. It is activated by dephosphorylation, which is favored by insulin. Citrate, a metabolic product of the Krebs cycle, also increases the activity of the acetyl coenzyme A carboxylase allosterically. Phosphorylation, which is triggered by the hormones glucagon and epinephrine, inactivates the enzyme and reduces its sensitivity to activation by citrate. That is, it does not remain sensitive to the citrate, which is it is activated, thereby slowing the fatty acid synthase process. Also, it should be noted that transition to the inactive conformation is associated with dissociation to yield the monomeric forms of the enzyme, that is, protomers are released. In the active, that is the dephosphorylated conformation, which is triggered by the hormone insulin, acetyl coenzyme A carboxylase associates to form the multimeric filamentous complexes. That is, complexes are formed in its dephosphorylated conformation. This antagonistic effect of the insulin produced when blood glucose is high is attributed to the activation of protein phosphatases. Not only the phosphorylation and the dephosphorylation of the acetyl coenzyme A carboxylase, but its activity is also regulated by local metabolites. For example, I would like to tell you first that palmitoyl coenzyme A, which is the principal product of the fatty acid synthesis, is a feedback inhibitor, not an activator, but a feedback inhibitor of the enzyme. Palmitoyl coenzyme A promotes the inactive conformation, and it diminishes the production of the malonyl coenzyme A, which is the precursor of fatty acid synthesis. Citrate, a metabolic product of the Krebs cycle, it also plays a central role in diverting the cellular metabolism from the consumption of metabolic fuel to the storage of fuel as fatty acids. Citrate allosterically activates acetyl coenzyme A carboxylase and thus is an allosteric activator of the enzyme, and it increases its V maximum. Citrate, as all of we know, that is adequate when acetyl coenzyme A is entering the Krebs cycle, when the concentration of metabolic mitochondrial acetyl coenzyme A and ATP increases. Citrate is transported out of the mitochondria. It then becomes both the precursor of the cytosolic acetyl coenzyme A and an allosteric signal for the activation of acetyl coenzyme A carboxylase. At the same time, citrate inhibits the activity of phosphofructokinase first and therefore reducing the flow of carbon through glycolysis. I would like here highlight the fact that this acetyl coenzyme carboxylase of plants and bacteria is not regulated by the citrate or by the phosphorylation dephosphorylation cycle. Instead, the plant enzyme, that is the acetyl coenzyme carboxylase of the plants, is activated by an increase in stromal pH and magnesium concentration, which occurs on illumination of the plant. That is, the acetyl coenzyme carboxylase is not regulated by the phosphorylation or the dephosphorylation cycle, but its activity is controlled by the stromal pH and the magnesium concentrations. After the plants, I would like to highlight here that bacteria also, they do not use triacylglycerols as energy stores. For example, in E. coli, the primary role of this fatty acid synthesis is to provide precursors for membrane lipids. The regulation of this process is but complex. It involves the guanine nucleotides that coordinates the cell growth with membrane formation. Dear students, I hope that today you have understood the lecture which was on fatty acid biosynthesis. Thank you.